So our first speaker, as I said, will be Dr. Stéphanie Longet, which I will have the pleasure to introduce. So during her PhD project at the University of Lausanne and her 5-year postdoctoral experience at the Trinity College in Dublin, she focused her work on passive and prophylactic oral vaccine strategies to induce protective immune response at intestinal level using in vitro and preclinical models. Then during her 4-year postdoctoral experience at the Public Health England and at the University of Oxford, she was strongly involved in characterizing systemic and mucosal adaptive immune responses in preclinical model in humans following SARS-CoV-2 infection or COVID vaccination. And in March 2023, she joined the University of Etienne as an assistant professor, and her current research interests are the development of nasal vaccine strategies against viral infections and the characterization of mucosal memory immune responses, including tissue-resident memory responses in the respiratory tract. And in addition to all of this, she is also co-organizing the French semester of the Live Erasmus Mundus Master, which is a program dedicated to vaccinology. And today she will share with us her journey, her European journey in the field of pharmacology. So I'm very happy to welcome her. The stage, the floor is yours.
Thank you. Thanks for the introduction and for the invitation. Just let me know, my network is quite unstable now. The connection is not good. So maybe I'm going to stop my my camera when I share my slide. I will see. But let me know if it's not good. I will change my internet network, but hopefully it will be fine. Okay, so I'm going to share the slide. Okay, can you see my slide?
Yes.
Perfect. Okay. So yeah, so thanks for the invitation and the introduction. So today I'm quite happy to share with you my journey in science and more specifically in the field of vaccinology. I would like to share, I would say it's my academic journey, but which is quite classical as academic path, I would say academic journey. The fact we move to different labs in different countries. So it's what I would like to share with you. I would give you some tips and maybe some advice. I'm currently assistant professor at the University of Saint-Etienne. So I'm more specifically in the Department of Pathophysiology and Biotherapy of mucosal infections. And in case you have my email address, if you have more questions after my presentation. Okay, so I'm going to summarize a little bit my journey. So my topic, my research topic, it's mucosal vaccination, and actually it was my topic during my whole journey. So basically, I started my studies in biology at the University of Lausanne in Switzerland. I did a master. At this stage, I was focused on genital immunity, and I work on Gardasil, basically. So in this case, it was not mucosal vaccines, but it was vaccines to induce mucosal immunity, so genital immunity. After I did a PhD at the University of Lausanne, at this stage, it was more focused on intestinal immunity, and I work on passive strategies by mucosal route. After I moved to Trinity College Dublin in Ireland for first postdoc, a 5-year postdoc, and I work on oral vaccination. So I work on the development of novel oral vaccines against bacteria. After this postdoc, I did another postdoc at UK HSA and after at the University of Oxford in the UK. And basically, when I started this postdoc, I wanted to change a little bit my topic. It was still about vaccines, but I wanted to work more on vaccines against Ebola virus and Lassa virus. But in 2020, as you know, SARS-CoV-2 emerged, and finally I work much more on SARS-CoV-2 infection and SARS-CoV-2 vaccination. And basically here I work on a novel field, because before I work on genital immunity, intestinal immunity, and now I'm more focused on respiratory immunity. So I took the opportunity to SARS-CoV-2, to COVID-19 pandemic, to explore another mucosa. And now I'm at the University of Saint-Etienne, and with my team, we develop vaccines by intranasal route, and we explore and we try to understand tissue-resident memory immunity. Just going to close this window. Oh, sorry. Sorry, there is an issue. Sorry, I stopped sharing. I wanted to close the window because I couldn't see my text. Oh, no, it's not working. Be fine. Okay, hope you can see my slide. Here you have just a few, on this slide you have just a few metrics related to my current position. Okay, so I'm going to go back to the different steps in my journey in science, and also the different challenges I had to face. So first, I did a bachelor in biology at the University of Lausanne and a master in genomics and experimental biology. In Lausanne, we have two master internships. We have a short one and a long one. So the long one, as I told you, I was in the field of HPV vaccination. I especially work in preclinical models, so in mouse models. And I decided to pursue in mucosal immunology and more in the field of intestinal immunity. So during my PhD, still at the University of Lausanne, I characterized antibody-based therapeutics, which can be given by oral route. And these therapeutics were against intestinal bacteria. So it was especially in vitro work. So I use an in vitro model, an in vitro intestinal model to test this antibody therapeutics. It was more or less a passive immunization in this case. It was in collaboration with a private partner in Switzerland, and it was quite interesting to collaborate with a private partner at this stage, because I had both sides, so academic side and private side. So it was quite interesting for me as a PhD student. At that time, I published 4 publications, so 1 during my master and 3 during my PhD at the University of Lausanne. So basically, after my PhD, I was thinking about my future, and I really enjoy working in academia. So I decided to pursue with a postdoc. But at this stage, you have several options to pursue with a postdoc. So you can apply for funding in order to do a postdoc with your own funding, or you can also apply for a fully funded position. But I decided to apply for funding, so I applied for a Swiss National Science Foundation postdoc fellowship to spend 3 years in a lab in Cambridge. I also tried to apply for an EMBO application, but both failed. So I was a little bit sad. They were my first grant application, fellowship application. So the first one, when it fails, it's quite sad. After, we are quite used to be rejected because it's like this. But I remember the first time, because it's a lot of efforts and it didn't work. So at this stage, after this failure, I decided to start my plan B. So the plan B, it was to apply for funded postdoc position in Europe. At that time, I decided to focus on Europe because I wanted to stay not too far from my family, basically. So I applied to more or less 10 positions in Europe. And finally, I got one, a 2-year postdoc position in Ireland at Trinity College Dublin in Ed Lavell's laboratory. So it's a laboratory focused on adjuvants we can use in vaccines. And they focus also on, at that time, I would say they were focused on oral vaccinations. So it was perfect in my field because I had some skills in intestinal immunity, I had some skill in vaccination, I had some skill in mucosal immunity, so it was just perfect. And at this stage, I work on a novel delivery system to administer oral vaccines and administer adjuvants by oral route. The first stage was in collaboration with a private partner in Ireland. So again, it was, sorry, I'm going to. So again, it was very interesting to collaborate with a private partner again, because we learn other strategies. It's not exactly the same strategies as in academia. So I thought it was very, very relevant to pursue this type of partnership. As it was, I would say at the time it went well. So I decided to apply again for a Swiss National Science Foundation postdoc fellowship, because in Switzerland we have the opportunity to apply for fellowships dedicated to Swiss citizens, but to work abroad. So I decided to apply for the fellowship and I got it this time. So it was a great opportunity to stay in the lab. I also applied side by side to Irish grant, Irish fellowship for postdoc. I got it as well. But at the end, when we got both for the same project, we have to decide which one we have to keep. And I kept the Swiss National Science Foundation postdoc fellowship because it was for 3 years and the Irish one was for 2 years. So I decided to stay 3 more years in the lab and I work on the modes of action of a specific adjuvant, specific oral adjuvant alpha-galactosyl ceramide, so alpha-gal-cer. And the fellowships basically cover my salary for 3 years. So it was great at this stage to get fellowships because when you start a postdoc, it's important to show you are able to become more and more independent, not only in the development of your project, but also how you have to fund, how you have to support your project. You have to show you are able to get some grants. So at this stage, I was very happy. So I was able to develop much more my research project. I also applied for other grant applications, but the other ones were not successful. I had the opportunity to supervise 5 undergraduate students, so I was able to supervise their own project, the lab work, the evaluation of the thesis. So it was very, very interesting to start supervising students. I also participated in national and international conferences. And as usual, I would say in academia, we have to write some papers. So at that time, I published 7 papers in 5 years. After this, so it's always the point with position in academia, you have always to think about the next step because usually the positions are not permanent. So you have to think about the next step. So as I was still enjoying working in academia, so the next step was to do another postdoc, because at this stage I was junior postdoc. So usually to stay in this journey in academia, we have to do several postdocs, not too much, but several. So for my second postdoc, again, I decided to apply for a fellowship. So the Marie Curie fellowship, so it's a European fellowship, it's quite competitive. And I wanted to do a postdoc at Serum Statens Institute in Copenhagen, because they are also expert in vaccines, in the development of vaccines. But basically it failed because my score was only 83%. So from 85, you have a certificate, and usually the project with more than 90%, but usually it's more 92, 93, depends. They are funded. So it didn't work. I also tried to apply for another fellowship, an industry academia fellowship, because I had the experience to work with a private partner. So I thought it was a good opportunity to try something a little bit between industry and academia. But the private partner decided not to pursue the project. So again, I activated my plan B. So I applied for funded position in Europe. At this stage, I applied for a lot of positions. 20, I submitted 20 applications in academia in my field. So I think it's very important to focus on applying for position in your field, because there is no point to apply for position which are not in your field. You won't be selected. So I focus on mucosal immunity and vaccination. So I applied for 20 position. I got 3 interviews. I also applied for position in the private sector, but I didn't have any interview. And I think it reflects what it is actually, because I always work in academia, so I have the skills which match very well with academic position, and probably a little bit less with position in the private sector, even though I had worked with private partners. Okay, so I got a position in the UK in Professor Miles Carroll's laboratory. So this lab was at UKHSA, and after the lab moved to the University of Oxford. So as I told you, I wanted to work a little bit in another field, but still field of vaccination. My project at that time was focused on immune response in Ebola virus survivors, and also seroepidemiology of Lassa virus. And we wanted to understand a little bit more immunity after Ebola virus infection and after Lassa virus infection, in order to develop, to optimize Ebola virus vaccine, or to develop vaccines against Lassa virus. But SARS-CoV-2 emerged, and finally I work on immune responses after SARS-CoV-2 infection and COVID-19 vaccination. So at that time it was very, very, very busy. So I work on different preclinical models and several cohorts of patients. So basically, at that time, it was a great opportunity, because I was able to be involved, write a lot of papers, and to be involved in a lot of papers. So at that time, I published in 2 years 39 publications. So it's really due to the fact there was the pandemic. So again, as senior postdoc at this stage, I had to develop my own research project. Again, I had to apply for grant applications. I had to apply for grants, and but most of them were rejected. I also supervised 3 undergraduate students, and I co-supervised my first PhD students. So I co-supervised the project, the lab work, and also the evaluation of the PhD studies. And I went to a couple of conferences, but actually after there was a lockdown and everything, so it was more complicated. So I participated in online conferences, especially. And finally, so after this, in 2023, at this stage, I did 9 years of postdoc. So at this stage, it was quite a crucial moment because I had to consider my future again. Because at this time, doing another postdoc and another postdoc and another postdoc was, I thought it became a little bit deleterious. It was good to do several postdocs for several years in order to develop my skills in academia, to show I was able to get my own funding, to supervise students, to publish and everything. But at this point, I wanted to evolve a little bit more and to try to have either a permanent position or a position which can lead to a permanent position. And actually, my goal when I started my journey in academia was to develop my own team. So basically, at this time, I considered several opportunities in the UK, but I wanted to stay in the field of vaccination and in the field of mucosal vaccination because it's really something I enjoy working on. And one day, the head of the department of GMAP, it's a French abbreviation for the department at the University of Saint Etienne. It's a department dedicated to mucosal immunity. So at that time, one day, the head of the department called me, and he told me there was a position like a lecturer position in the department. So he asked me if I was interested. So I took 1 month to decide what I wanted to do, because at that time I was at the University of Oxford, but in the same time, there was no opportunity in Oxford and no opportunity in the UK in this field, and for me, I would say. So I decided to take the position, and after I moved to another position, tenure track assistant professor position. It's quite new in France, this position. So it took some time to consider if it was great to take this type of position. But I decided to take it. So it's a 3 years position. It's not permanent now, but it's a tenure track. So if everything goes well, now in 18 months, I will have the tenure position. But I thought it was really a great opportunity. I don't regret anything because it really boosted my academic journey, because now I'm supervising 5 PhD students, 1 postdoc. I supervise also students dedicated to give lectures. It's something probably specific specific in France. And I'm also supervising master students. In the same time, with this type of position, we can get some specific funding. It's funding to cover salary, so you can recruit somebody. So I recruited a postdoc because I had already some PhD students. So yeah, I'm very happy to have this position, because now I can develop a new project. Now my daily work, I would say, changed a little bit. Before, I was doing a lot of experiments, bench experiments, wet lab, I would say. Now less and less. There are more my PhD students and my postdoc who are working in the lab. So now I have to write a lot of grant applications, because we need to make sure to have enough funding to cover everything in the lab, the work in the lab. I have to supervise the project and my team. I have to develop collaborations, to attend national and international conferences. And I also part of a lot of juries. So it takes quite a lot of times now. So basically, I would say, when I just consider my daily work now, I write a lot of grant application. I write a lot of papers as well. And I'm part of juries, I would say. And so just to finish with this, so I'm very happy because actually it was my goal to reach this type of position, to have my own team. It's quite challenging. Actually, every step is challenging. And I would say it's more and more challenging. But I'm quite happy because it's what I wanted to do. I wanted to have my team. I wanted to develop some project and to supervise the team. So it was an exciting journey, but sometimes, of course, it was quite challenging. The fact to move abroad, it can be quite challenging, especially for the first postdoc, because for me, it was the first time I was leaving Switzerland. But in the same time, it's quite great to discover a new culture. It was also great to improve my English. By the way, I decided to focus on English-speaking countries for this reason, to improve my English. But at some point, I was ready to move elsewhere and to learn a new language. So I think it's important to be very, very flexible and open to everything at some point. Academia is quite great because you have quite a freedom. You can develop the project you want, more or less. Even so, you have to match with the goal of the department, of course, the goal of the universities. And I would say you have sometimes hot topics, so you have to align your project with the hot topics in order to get some grants. So, but yeah, it's what it is. You have also a pressure to produce results and to publish. Sometimes it's rejected. Some publications are rejected, so you have to find a plan B and to work again in your project in order to improve your papers. Regarding grant applications, it's also a lot of work, a lot of effort, and most of them fail. It's not personal. It's for everybody. It's like this. So you need to be persistent and to continue and to try to improve your grant applications. And at some point, it works. And in academia, it's quite difficult to get a permanent position. And actually, my first permanent position should be in now 18 months, 15, 18 months. So you see, you have to live like this with 2 years, 3 years position, and again you need to take some time to think about your future. So yeah, it can be challenging sometimes. So you have to be passionate about science because especially in challenging periods, if you don't have the patience, I think you will just give up probably this type of journey. Something which was quite important for me is to understand my role in the society, and during the pandemic, it's something which was very, very important to me. I could also meet people coming from everywhere, the fact to live in other countries. And now I have a team which is very international. So I have people from China, Russia, India. So basically, it's something which was very important to me to develop a very international teams. And it's probably due to the fact I travel a lot for my postdocs. And I think it's also important to be curious and creative in order to be successful in this journey. I think time is flying quite fast, so I'm going to switch to the master life. So another opportunity I have now at the University of Etienne, it's to co-organize a very fantastic master leading international vaccinology education, so master live. So there are people who basically the applicants come from everywhere, so it's very international, and the reason why I really enjoy co-organizing this master. And I think it's more and more needed to have future vaccinologists, very skillful vaccinologists, and it's the reason why this master was developed a few years ago. So basically, this master, it's Erasmus master. So you have basically it's funded, it's organized by by 5 full partners. So you have partners in Barcelona, partners in Antwerp, and in France, so Lyon and the University of Saint-Etienne. Here you have the flyers, because now we are recruiting the new students. Here you have the links. So I would like to give you just a few information about this master. So regarding the organization of this master, so you have Master 1 and Master 2. Most students are included in both Master 1 and Master 2, so it's a 2-year program. But we also accept some M2 students as well, only. So these students only are involved in the master only for 1 year. So you have 1 semester in Spain, 1 semester in Belgium, 1 semester in France. And the last semester, it's for the internship, and it's worldwide. So the first master is focused on immunology, immunopathology. In Belgium, it's more focused on infectiology and vaccinology. And in France, again, it's also focused on vaccinology. You have some teaching unit focused on vaccine formulation, vaccine-specific application, also communications in the field of vaccines. And there is also a practical session at the University of Saint-Etienne. After this 2-year master, the students who complete the full 2 years receive actually 3 accredited national master degrees jointly. And you have the master from the university, from Barcelona universities, the master from Antwerp, and this one from the University of Saint-Etienne and University Claude Bernard in Lyon. Regarding funding, so this master is funded by EU, and the EU can give some scholarships for the most for the best students. So here you have this type of fellowship students can have for 24 months. But there are also other ways to fund, to support this master. So some funded can be found via associated partners, and you have also other ways. It can be also self-funded. So again, I show you the leaflet. You can see there is a new deadline. So the deadline for this year is the 21st of January. So the applications are still open. And you have here the QR code and also the link if some of you are interested in this. And actually the deadline is always mid-January. So it can be also, you can also apply another year. Okay, so with this, I finish with my presentation. So I'm going to stop sharing and turn on my camera.
So thank you very much, Dr. Longet, for this very interesting talk, and also thank you for being so transparent and vulnerable about all of the challenges that you encounter during your journey. So I think there are no questions for now from the chat, but I just had a quick one before we move to our second speaker. So you really insisted on the importance of staying in your field and really digging deeper into that one field, and yours was mucosal immunity. And I wanted to ask you, what is the most interesting or passionate thing about that field for you, and why you chose to keep pursuing it?
Sorry, my internet connection is not so good. So the question was why I decided to pursue in the mucosal immunity, mucosal vaccination field. Is it correct?
Yes. Why that specific field exactly?
I cannot hear anything. Sorry. My connection. Yeah, okay, sorry. Yeah. So I would say vaccination first. It's when I was teenager, I thought vaccine was very fascinating field because it could prevent the disease. I think for me, it's fantastic to prevent a disease instead of waiting for the disease and to try to treat the disease. So for me, it was a fantastic uh, way to prevent the disease. So when I was teenager, I decided to work on the field of vaccines. Why mucosal immunity? It became interesting a little bit later. Basically, I discovered mucosal immunity and mucosal vaccine a little bit later. For me, mucosa are very fascinating tissues because you have a lot of interactions between the environment and between our own body. So it's quite complex, and the complexity, I was fascinated by the complexity of this type of tissues. And I was, wow, it's fantastic if we could give some vaccine by this route in order to have in these tissues. So it's the reason why it's really the topic. I was fascinated by the topic, and it's the reason why I decided to pursue in this field.
Thank you very much. So now we will move to our next speaker. But again, if you have questions for Dr. Longet, please ask them in the chat. So our next speaker is our early career researcher, Ameyo Daglus. So he's an early career vaccinologist with an Erasmus Mundus live master's degree. So really in line with what Dr. Longet talked about. So his work and thesis focused on next generation mRNA vaccine engineering to achieve improved antigen expression, stability, and immune activation. He is also a passionate advocate for cancer vaccinology and equitable vaccine innovation in Kenya and the co-founder of Kenya Vaccine Foundation. He is also an active member of Kenya's Society of Hemato-Oncology, the Royal Society of Tropical Medicine, and the American Society for Microbiology. And currently, he is seeking PhD opportunities in immunology or RNA sciences. He is deeply engaged in mentorship, health communication, and community-centered innovation. So today he will share with us his work on the characterization of monocistronic and polycistronic sequences for the development of an mRNA vaccine. Ameyo, the floor is yours.
All right, and thank you for the wonderful introduction. So I'll share my screen right away. Okay. Can you see my screen? I'll put it in presentation mode. All right. So I'll start the presentation right away. My name is Ameyo Daglus, and Happy New Year. I'll be glad to do my presentation today on advancing mRNA vaccine engineering to enhance antigen expression. So I'm a recent graduate of international leading vaccinology education and Erasmus Mundus Master. So just in a nutshell, I'll discuss about the introduction. I'll introduce their mRNA and then discuss a little about their construct designs, the mRNA synthesis, protein expression, and lastly, a little bit about their Erasmus experience. So just to start with, and we do remember that during COVID-19, new technologies and even new method for development of vaccine came up, and mRNA was one of it. And we saw it was like a really great breakthrough that the development was quite fantastic and even over a short period of time. And due to this, over time, different groups, they have come up with different mechanisms to improve even on this technology and grow even the growth of this kind of vaccines across in cancer, in allergies and so forth. So basically, it will be important for us to see the structure of an mRNA or discuss what is actually an mRNA for those who are not in this field. So an mRNA is usually have the cap, the 5' UTR, the antigen or allogen in this case, the 3' UTR and poly A tail. And all these parts are actually very important for the translational efficiency and the stability of an mRNA. And once you have your mRNA construct, it's usually encapsulated in lipid nanoparticles, which are usually used as vaccine in this case. So basically, there are different mechanisms in which we can see how the mRNA works. In this case, it's in once you want to induce tolerance or for inflammation or hypersensitivity. So what actually happens is once you have been injected with an mRNA vaccine, the lipid nanoparticle usually releases the mRNA, and then it is usually translated in the ribosome, and then depending on its nature, if it is freely secreted, intracellular or membrane anchored, then it is usually released. So with this, it elicits immune response whereby it can be identified by the immune cell, either by histocompatibility complex 2 or BCR, that is B cell receptors, and in situation of allergy or inflammation, this activates TLP1 and TREG, which then balances TLP1 and TLP2, which is really crucial for controlling inflammation and all that. And the same happens with IgG4 activation, which we know is very great in controlling inflammation. So with this kind of technology, you are able to induce tolerance, and this can be memorized again and again, which is really fantastic. So just to focus on the different engineering to have different constructs, either monocystronic, that is means encoding for a single protein, or polycystronic, in case if you're targeting or you're making a multivalent vaccine. So in this case, I was actually able to work on a protein that has 3 main variants, and I anonymized them as protein 1, 2, and 3. And this protein, they had 76% sequence homology between themselves, and they had between 396 to 398 amino acid with a signal peptide, unstructured region, and beta sheets. So with 19 construct, so 13 of them, they were monocystronic. That means they were encoding for a single protein. And out of these 13, 11 were actually just encoding for a single protein, but 2 of them, they were encoding for polyprotein. That means taking either one protein 1 and 2 and mixing them, and then at the end you just have like one protein. Or for polycystronic, which were the 6, 4 they were linked with the 2 peptide linker, and 2 of them they were linked with the iris sequences. So these are just internal ribosome entry sequences, and we'll see as we progress what was the impact for each design in this case. So, and one of the bigger question was, why is it even necessary to have so many different engineering or designs? And this is just simply because of the differences in the translation mechanism based on the designs. So as we can see here, for monocystronic constructs, everything happens in one open reading frame where translation happens at the cap, then your protein of interest is translated. And that is as simple as that. But for polycystronic, when you have 2 peptide linkers, so the first protein is usually translated. And then after that, in the 2 sequence here, there is ribosome skipping that happens between the proline and glycine. This is because these 2 amino acids, they're unable to form actually a strong peptide bond, and then the second protein is again translated. So at the end, you have to translated protein, but everything happens in one open reading frame, as we can see. And this is quite different for polycysonic mRNA with an IRS sequence, as we see that the first protein is cap dependent, and then after that, the second protein is IRS dependent. So this basically shows that there are two open reading frame, and at the end, you have your two separate proteins well translated. So having this in mind, so the concepts, the designs that we had here is that the first three proteins, they were original without any modification. And then here, there is the deletion of the unstructured region from this part, and then the deletion of the signal peptide here, and then the replacement of native signal peptide with a viral one, and then the addition of viral transmembrane domain. And again, sorry, this was just important to note that with this addition or removal of the signal peptide is for the localization of your protein. Either if you want it to be freely secreted, you're going to have the signal peptide, and then this one will be intracellular due to lack of the signal peptide, and then this one is membrane anchored due to the addition of viral transmembrane domain. And for the monocystronic construct that they were encoding for polyproteins, two protein, that is either protein 1 and 2, they are linked with a simple amino acid, in this case serine and glycine. And the difference between these constructs, the construct 1 and 2, is just the different number of the amino acid, but they are made up of the same amino acid, that is glycine and serine. And these were chosen because they are flexible amino acid, they're simple amino acid, I mean. And for polycystronic constructs, so either protein 1 and 2, they are either linked with 2A peptides or IRS sequences. And just to note that these 2A1 and 2A2, they are actually from different viruses. So that is why it's 2A1 and the same here, IRS1 and IRS2. They are the construct or the sequences they are obtained from different viruses as well. So with this kind of design, you are able to have linearized DNA fragments, your starting material, to be able to check if your construct works. And this happens by in vitro transcription. So what happens basically with your linearized DNA fragments, you are able to generate your transcripts, and then with your mRNA transcript, you can carry out cell-free translation. You use a cell-free kit and fluorolysin for visualization, and then you can visualize if your design actually worked with the SDS purge. And if you want to quantify you with the cell-free translation as well, you carry out HPLC-MS, and you'll be able to quantify and be able to identify if your design for the construct actually worked. So just to show some of the expression after with the cell-free translation, it was actually interesting to see that the constructs that that were from this, that were encoding a single protein, that were monocystronic. So if you can see here, this is protein 1, protein 2, and protein 3, and this is codon optimization 1 and 2, as everything was done in double, just to double check that it works. And we can see that the expression was as expected, and this was really fantastic to see. So after this, we checked for the monocystronic constructs that they were actually encoding for polyprotein. And this basically means here are the controls for protein 1 and protein 2. And these are polyprotein. That means you have protein 1 and protein 2 mixed together with serine glycine linkers. And the difference between this construct and this one is that the difference in number of amino acid for the linkers. So this one had 27 amino acid, and this one had 45. And actually, this is what was expected. For the polycystronic construct, it was slightly interesting to see that the ones with 2A peptides here, the first bands here with the SDS purge, they were much brighter compared to the second one. And this basically means that the translation was not as perfect as expected. And this was different with the iris linkers, as we could see that here. For this iris one, both bands, they were brighter. That means the translation was as expected. And then for the iris 2 here, it didn't work because we could only see the first bands. And as you said, that the second band is iris dependent, and this basically doesn't mean didn't work. So just to summarize for the rest of the construct, because it was constant for this, that all the monocytonic construct that they were encoding for a single protein, they were actually well translated as expected, and also those that were also encoding for polyprotein, they were as well expressed as expected, as we could see. But the one that had to a peptide, they were not actually well translated as expected. But again, these should be and could be much optimized and tried again in the cells, might have different expression or results. And then here for the iris, the first one, iris 1 worked perfectly, but the second one didn't. So at the end, I selected few transcripts for protein quantification. And again, just to show that how this works for the protein that was quantified, it was interesting to see that protein 2 actually, regardless of deletion of the unstructured region or deletion of the signal peptide, it was well expressed compared to protein 1 and 3. And then again, it was interesting to see the polyprotein that had a linker with 27 amino acid was much better expressed compared to the one with 45. So this was really interesting to see that these kind of designs, they are really important, and they help in designing polyproteins that can be used for multivalent vaccines. So with just this little bit representation, it was actually pertinent and important to see that polycistronic construct and polyprotein construct can be used for design of multivalent vaccine. And of course, this kind of work is much used and useful for much design of multivalent vaccines. And we hope that in the near future, there will be fewer shots and greater impact. So yeah, for this work, I'll stop here. And again, just to emphasize on what Dr. Stéphanie said about the Erasmus as being the recent graduate of the Plotkin promotion, I'll just share a little bit few insights, and I'll not repeat. Actually, I think most of the things she said is actually the most experience about it. But again, I'll share just a little bit of the experience that I had as part of it. So just to start with is that each year, this amazing and fantastic program usually recruits at least or about 30 seats. That might be slightly more or less, and they have around 20 to 25 full scholarship. And the amazing thing about it is that it takes a quite diverse field here, as you can see. So we have medical doctors, pharmacists, veterinary doctors, and scientists in the class. So it's kind of very international. Anybody, everywhere in the world, you can apply, and regardless of your field. And it is really kind of very an amazing experience to be in this kind of pool of training. So as I won't repeat this, actually for my experience, I was directly selected to Master 2, so that is 1 year. And this is actually possible if you have an experience in immunology or microbiology and at least 240 ECTs. Otherwise, you have to go through the mobility Master 1 and Master 2, as Dr. Stéphanie had explained. And again, one of the key and an amazing thing is that with this program, you are actually having the ability to have a direct connection with great scientists, and again to visit one of the best amazing institutions, Sanofi, VFI, CEA, and all that. And again, you can choose to go anywhere in the world for your internship. So I really encourage all the interested students or researchers to really apply this. So I won't repeat this, but if you go through the application and you need you have more inquiries or something, I'll be glad to help as an alumni, and even for the seniors who might have positions for the PhDs or research assistants. So this is my contact detail. You can contact me, and I'll stop there and welcome any questions. And thank you so much for listening and for this opportunity. And I'll stop sharing.
Thank you very much, Ameyo, for this very interesting talk. So there is one question from the audience. I don't know if you have already covered it. Maybe I missed it, but they're asking which platform was used, if you can share, for the translation of the vaccine constructs.
Yeah, all right, and thank you for that amazing question. So this was by use of bioinformatic tool, and in this case we use Genius to redesign these constructs.
Thank you. I also had one question. You mentioned multivalent vaccines as one of the perspectives for this technology. Do you have any disease or specific application in mind if you were to pursue this project, or maybe that you had specifically starting it with?
Okay, yeah, thank you for that question. So in this specific project, this design was specifically for inducing tolerance in allergy. So I mean, like a specific allergy, but because of this work is still into development and stuff. So at the point I can just say like a specific allergy, but with the same design and the same knowledge, this can still be applied to infectious diseases and even in cancer.
Okay, thank you very
Yeah.
much. There's a question also for Ameyo from Nina Nguyen from the State and Serum Institute in Denmark. And she's asking, how is it possible to direct regulatory response rather than a pro-inflammatory immune response using this mRNA vaccine? And specifically, if this is achieved through the choice of the adjuvants, the modification of the mRNA itself, or the inclusion of other immunomodulatory molecules.
Yeah, thank you so much, Nina. That is a wonderful question. So basically, if you want to achieve if I go to your question, if you want to achieve a direct regulatory response, so this depends with the kind of antigen that you're using, and of course, if you're using like lipid nanoparticles, so some of them with the improvement, they're actually being designed to be more specific, depending on the type of immune response that you're targeting.
Okay, and yes, there's another question for you, Daglus. How can, and if possible, these vaccines target dendritic cells?
Okay, yeah, that's a good one. Of course, depending. So basically, for my first design, it was much based on tolerance. But of course, if again you want to apply it for maybe cancer and all that, of course, it is something um that can be actually be done. But of course, it's not one of the things that I really looked into, but it is actually possible to make these kind of designs as well.
Great. And I had a question for Dr. Longet on the mucosal immunity field. What is the place of mRNA vaccines, or is it something that your lab is also investigating, or not at all?
I think she left.
I think Professor Longet unfortunately had to leave us. So yes, but I saw that she answered a lot of the questions that was addressed to her in the chat. So there's no other questions. So I would like to yes, I just saw that she said that she just left. So as there are no other questions, I would like to warmly welcome both Dr. Longet and Ameyo Daglus for these two very insightful talks. Our next gen webinar will happen the 5th of February, featuring Dr. Isaac Sakala from the Centenary Institute in Australia and Dr. Laura von Mertel from the Founders Institute for biotechnology, and it will be a session dedicated to tuberculosis vaccinations. So more information will come very soon. Thank you very much for joining this first webinar of the year, and I wish you a very pleasant day. Bye-bye.